So the hormones produced by the plants are known as phytohormones, and they have also a profound effect at very low concentration. Okay, there are many signaling molecules which help the plant in resistance against the pathogen, defense against pathogens. They include jasmonic acid, salicylic acid, and the polypeptide cysteamine. We will also study all these signaling molecules when we will be doing the stress physiology chapter. So today we are going to deal with auxin, gibberellin, cytokinin, ethylene, and abscisic acid and other growth regulators. Among these other growth regulators, I will be dealing with brasinosteroid in the plant hormone chapter, and the rest of the hormones I will be dealing in stress physiology, or I will do it separately. Now we have to understand and we have to remember also the different precursors of plant hormone. Okay, how the hormones are synthesized. The biosynthesis part is also there in our syllabus, so we have to focus on it, and part B questions come from here as well. Okay, so IAA, which is the first hormone discovered, named as auxin. This auxin is synthesized from the from the amino acid named as tryptophan. The another hormone ethylene is synthesized from methionine. Now another hormone, cytokinin, gibberellin, strigolactone, abscisic acid, and brasinosteroids, they are synthesized from IPP. The full form of IPP is isopentenyl pyrophosphate. Okay, and the pathways which use isopentenyl pyrophosphate, these pathways are known as isoprenoid pathway. One another signaling molecule named as jasmonic acid, they used alpha-linolenic acid as their precursor, and the nature of this alpha-linolenic acid is a lipid. Okay, now these you have to remember. So a small question I have embedded, that the plant hormone indole 3-acetic acid is present in most plants. The structure of this hormone is related to which of the amino acid? Now IAA, since auxin is derived from indole compound, the name of the auxin is indole 3-acetic acid. Okay, and the precursor of IAA or auxin is tryptophan. So the answer will be tryptophan. Now senescence. So let's understand about what is the what is the meaning of the phenomena senescence. So senescence is a process in which the plants show several kind of symptoms. For example, they shed their leaves, they form abscission layers. Okay, why do they do so? To protect themselves from a certain kind of environmental condition, to protect themselves from any kind of stress. The extra organs they shed, okay, they form an abscission layer to separate the unwanted leaves, to separate the diseased plant from themselves, okay, the diseased organ from the main plant. Okay, now there is a difference between the term aging and senescence. Okay, as the name says, aging is a term when a plant attains a certain, so the spelling is wrong, sorry, I beg your pardon. So aging is when a plant attains a certain age and then they show these kind of symptoms. Okay, but senescence is, this can happen at any stage of the plant's life cycle. When they have to cope up with any kind of stress, they show senescence. Okay, but aging is when they attain a particular age only, then this happens. Okay, and then they cannot be saved. Okay, the plant dies after a particular age. Okay, now there are some hormones which does not allow the senescence to occur. These hormones mainly play the role in growth and development. What are these hormones? They are auxin, gibberellin, and cytokinin, which helps in the growth and development of the plant. Now, there are some hormones which positively regulate the senescence. That means they help the plant to show these kind of symptoms. What is the name of that? ABA, ethylene, brassinosteroids, trigolactone, jasmonic acid. They help the plant to show the senescence. Now starting with the first hormone, auxin. As discussed that this is the first plant hormone discovered and this is derived from the indole compound. So the name came as indole 3-acetic acid. In short, we'll say it as IAA. Okay, so if IAA is written, think that this is auxin. They are mainly produced in stem tip. They help in cell elongation. They can be naturally or synthetically derived. So they are mainly produced in the apical meristem. So apical region can be the root apical region and the shoot apical region. So this is the shoot region. So this is the root region. Root also have the apical region, this one, and the root also have the apical region. Okay, so they are synthesized in the apical meristem of both root and shoot. They are synthesized in all dividing and growing tissues, say for example shoot and root apical meristem, developing fruits, seeds, which are the primary site of IA synthesis. But the main site where most of the IA is synthesized is the shoot tip. Okay, main role is cell elongation. Now there were several experiments conducted to prove the nature of the auxin, where are auxin synthesized, what it does. Okay, so first experiment was conducted by Charles Darwin and his son Francis Darwin. The specimen used by them was canary grass, and this canary grass belongs to a family of plants known as monocotyledonous. Okay, so all grass family of plants belong to a group named as monocotyledonous. Okay, now these monocotyledonous plant, when they are very young, okay, in the young stage they are protected by a covering named as coleoptile. Okay, in the very young stage their shoot is covered by coleoptile. Okay, so if this is a coleoptile and here is a shoot inside, okay, so this region, which is a shoot of monocot plant, they are covered by coleoptile when they are in the young stage, and this coleoptile is very sensitive to light. Okay, now what they did, the normal experiment which is known to us, that is tropic movement. Okay, so what is trophic movement? Geotropic, phototropic. Okay, there are different tropic movements. Now, tropic movement is what? Tropic movement is growth towards a particular stimulus. Now, remember the term I am using, growth towards a particular stimulus. So, if it's phototropic, what we see? So, if it's phototropic, what we see, the bending of the light or the growth of the shoot towards the source of the light. Okay, the normal experiment, the basic idea which we all have, that when light falls on one side of the shoot will grow towards the source of the light. That is the growth towards the source of the light. Okay, so this is the general experiment. In the second experiment, he says that since auxin was synthesized in the shoot tip, okay, since auxin is synthesized in the shoot tip, okay, and when this region is illuminated or when light falls from any one side, say here I have drawn from the left side, if light falls from any one side, the plant will bend towards the source of the sunlight. Okay, light is here and the plant has bent towards the source of sunlight. So what is happening, that auxin which is synthesized here in the tip region, it moves downward. Okay, now this auxin when moves downward, it helps in the growth of this region. Okay, so this, I'm very sorry, I should draw it towards the left side, towards the another side. So see, Auxin light, the source of light is here. Okay, the source of light is here, and the bending is occurring towards the source of the light. The shaded Auxin was synthesized here. This is Auxin. Again, I'll change the color. It's taking time, but it's necessary for you to build the basic. Okay, so the green color, I'm representing it with Auxin. Okay, and the blue color is the source of the light. And I'll change the color for the shaded region as well. So see, this is the shaded region which is not receiving the light. So Auxin from here will move downward from in which region? Towards the shaded region it will move downward. Okay, towards the shaded region. See, one part is sunny region, one part is a shaded region. From the tip it will move downward towards the shaded region and will allow the growth of this shaded region. Okay, Auxin moved from the tip part to the shaded region and allowed the growth of this shaded region. Okay, so the shaded region had bent towards the source of the light. Okay, so to prove that this Auxin is synthesized the tip only, what he did, he covered the part below the tip to prove whether something is synthesized at the tip only or not. Okay, so if an opaque cap was placed on the tip, what happened? Light cannot pass through the opaque cap. So here the tip part, so Auxin here is not synthesized because the tip part is covered by the opaque cap. Auxin is not synthesized here, so there will be no bending. Okay, so when the tip part is remain unexposed, but the part below the tip part is covered with the opaque cap. Okay, so here the light is there. Okay, the tip part receives the light and the Auxin is diffused towards the shady region from the tip region downward towards the shady region. Okay, and this part has shown the growth. Okay, so the bending occurs towards the source of the light and the growth occurs in the shady region. Okay, so see, if the source of the light is vertically up, okay, what will happen? Auxin from the tip will move down. Auxin from the tip will move down. Just a moment. Auxin from the tip has been moved down. Okay, and stimulating the growth of the root. Now here what we see that when the light falls from one side, what will happen? The bending should occur towards the source of the light. Okay, so here the Auxin should be activated and should diffuse toward the shady side. See the dots here, you can see that the Auxin is diffused to the shady side. Okay, and helping in the growth of this shady region, Auxin is moving in this direction from the tip to the base. Okay, and this is known as, and here this Auxin is moved from the shoot tip towards the base. Okay, and helped in the growth of the shady region, because of which the shoot has bent towards the light. Okay, this, see, the shaded region has more Auxin. Okay, so where the region wherever the Auxin will be more, the cells will elongate in that region. Okay, so the main role of Auxin in is cell elongation. So wherever the auxin will be more, the cell will be elongated more in that region, and the growth will be more in that specific region. So look here, this experiment, see here the picture shows the experiment by two different scientists. First is Darwin and Darwin, second is by Boyson and Jensen. In this first experiment, what they show that the shady region elongates. Okay, see the shady region, the cells of the shaded region has more elongated cells because they have high amount of auxin. Okay, so auxin diffuses from the tip part to the shaded region downwards. So that, see, the shaded region has elongated cells and the illuminated side have the small number of non-elongated cells. Okay, so what experiment they perform if they have removed the tip part and allowed the light to pass through it. So the tip part is when removed, the auxin will not be synthesized, so no bending. Tip part removed, so no bending. If the tip part is covered with the opaque cap, light cannot pass through the tip part, so auxin cannot be synthesized, no bending. But if the tip is transparent, so light can pass through it. Okay, so the tip cap is transparent in this case, in this experiment, so light can pass through it, so bending occurred. But if any region below the tip is covered, but the tip part is unaffected, so light again can pass through the tip part and auxin diffuses downward and still shows the bending. Now another experiment carried out by Boyson and Jensen, what they proved that they have taken two separate material. In the first experiment they have taken gelatin. In the second experiment they have taken mica. When they have taken gelatin, what they did is that they have taken the polyoptile. Okay, they have cut the tip of the polyoptile, they decapitated it. The correct term of cutting the tip is decapitating the tip. Okay, then he took the gelatin block and placed the gelatin block in between and the tip back, and then allowed the light to pass through. It was seen that the normal bending occurred with the gelatin block in between. So what is the role of gelatin block? So even if gelatin block is there, the auxin can diffuse downward. Okay, even in the presence of gelatin block, auxin can pass through the gelatin block and then can diffuse downward towards the base. So what is the role, like what he wants to prove with gelatin? That the gelatin is here a water permeable substance. Gelatin is a water permeable substance which will allow the auxin to diffuse through it. That means auxin is a diffusible material. Okay, as it is passing through the gelatin block also. Okay, and a response is seen. That means gelatin is water permeable and auxin can diffuse through gelatin also. But when he placed mica in between, he conducted the same kind of experiment, but instead of gelatin he used mica, and he has seen that no bending occurred. Why? Because mica is water impermeable. Okay, so water impermeable substance will not allow auxin to diffuse downward. Okay, it will block the passage of the auxin from the tip to the base. So in that case, no bending will occur. So this experiment is to prove that auxin is a diffusible substance. Okay, mica is not allowing the diffusion to occur, but gelatin is allowing that. Thus, the conclusion of this experiment is that some signal is produced at the tip which travels towards the growth zone and causes the shaded side to grow faster than the illuminated side. So, I mentioned the growth zone. So, where is the growth zone? So, the region which is responsible for bending of the polyoptile towards the light is known as the growth zone, which is a region below the tip. Okay, the growth zone is the region located below the tip, which is responsible for the growth. Okay. So the signal is produced at the tip, it travels to the growth zone and from where the growth takes place. Okay. Now second experiment was conducted by the scientist named Wendt. He used the specimen Avena sativa named as oat, and the name of his experiment is known as Avena coleoptile curvature test. So look carefully at these experiments. They are done by different scientists. So Darwin and Boyson and Jensen were discussed. The conclusion of Darwin's experiment in Boyson and Jensen's I will discuss. You go through these slides. We'll discuss about PAL. So here what we see, when the tip is removed, okay, and the tip is placed onto one side only, okay. So say for example, this tip is already activated in light, okay. This tip, listen to me carefully, this tip is already activated in light and then it is removed, okay, and it's placed onto just one side of the coleoptile. When it is placed on one side of the coleoptile, what we will see, that the bending occurred. Now the bending will occur in which direction? So bending will occur wherever the auxin, the growth will occur in the region where the auxin will be high. Okay, so if the tip part is placed to this region, so the auxin concentration is high here. Okay, so the growth has taken from this side. Okay, so even in the absence of the light, this bending has occurred because the tip is already activated. Okay, so if I reverse the experiment, if I keep this activated tip part over here, so auxin is here, okay, in high amount is auxin should be here, and the bending will occur towards the opposite side. Okay, this will become the shady side and this will become the active side. Okay, this will become the shady side and this will become what, the sunny side. Okay, so the bending will towards this region. Okay, so now there may be one confusion that, wait, so now there can be one confusion. Explaining, just a moment. Okay, if light is from, okay, if the tip part is placed towards this side, then auxin concentration is high over here and the tip and the bending will be towards this side from the opposite side. Okay, now there might be the confusion that auxin diffuses from the tip region to the downward region. Okay, so auxin can diffuse. So here what we are seeing, that the bending occurred in absence of light. Okay, that means here there is no light source. Since auxin is diffusible, how auxin will decide where this is like the shady side or this is the sunny side? Okay, so auxin can diffuse from the tip part anywhere in the coleoptile. It can diffuse anywhere. This direction will not be limited to the shaded region, okay, and to the sunny region. So whenever the tip is placed on the coleoptile, when the activated tip is placed on the coleoptile, the auxin will diffuse everywhere in the coleoptile. But wherever the concentration is high, okay, the cells will elongate in that region. So what I would like to stress upon is that the auxin, since it is diffusible, this can diffuse to the entire region of the coleoptile. Okay, so doesn't matter where the tip is placed towards the right side or the tip is placed towards the left side, doesn't matter. Okay, since auxin is diffusible, auxin can diffuse everywhere, but there will be the difference in the concentration. Okay, in the shaded region the concentration will be more. Okay, but if there is no light, then what we will see? If there is no light, what we see, wherever the auxin concentration is more, wherever the auxin is more, in that region the cells elongate. I hope I'm able to make you understand this thing. In that region the cells will elongate. So what I mean, that even if the light is not there and the tips are previously activated, then when whichever region, now when the light is not there, there will be nothing to be labeled as shady and sunny. In that case, wherever the auxin concentration is high, that region will show the growth, because that region is able to elongate their cells. Okay, this is the experiment conducted by Paul, which shows that the auxin, since it's diffusible material, so in any direction when it is placed, it shows bending. Okay, so even in absence of light, so even in absence of light, what happens when the tip is placed either towards the left side or towards the right side, in whichever region the auxin is more, in that region the bending will occur. So the tip is previously activated, okay, and in whichever region the auxin is more, that part will show the elongation. Okay, now why in absence of light? Because the tip part is already activated. Okay, now I'll again conclude. But what Wendt did, Wendt took an agar block. He removed some tips from the coleoptile. Okay, he has taken few tips from different coleoptile, and what he did, he placed those tips onto agar block. Okay, now agar block can retain the diffusible auxin. So what happens when the, if some illuminated coleoptiles, okay, they have taken some illuminated coleoptiles. Illuminated coleoptiles means auxin has started synthesizing there. Okay, then they cut their tips. So what happened? What they did? They took some illuminated coleoptiles. I will draw it in a white page. So what he did, he took few of the coleoptiles, okay, he illuminated those coleoptiles, okay, so the auxin started synthesizing and he decapitated those. When he decapitated, he used that tips and placed onto the agar block. Okay, now since the tips were activated, what will happen? Auxin will diffuse onto the agar block. Auxin will diffuse onto the agar block. It will come out from the tip and diffuse and will retain on the agar block. Okay. Now this agar block has been cut into equal pieces, few equal pieces. Okay. And one of this piece is placed onto decapitated coleoptile. Decapitated means the tip was previously removed. Okay, and one of it was placed on decapitated coleoptile. This is what? This is decapitated. Okay, it was when placed on decapitated coleoptile, they also shows the bending. So what is the conclusion? That this coleoptiles releases the auxin on the agar block and agar block retains it. So when they are placed onto the decapitated coleoptile, the auxin diffuses from the coleoptile to the downward region. So whichever region has higher amount of auxin, that region will bend. Okay. So this was the experiment performed by Wendt. So look here, agar block placed on one side and bending occurs. So no light. Okay. So this says that in whichever region the auxin is more, that region will show the cell elongation. So see, in this region the auxin concentration will be more. So this region elongated. Okay, and as a result the bending occurred or the growth occurred from this region. Okay, so this was the experiment performed by Wendt. So this also happened in absence of light, because the tip part which was activated, the auxin had been started synthesizing in the tip part, and auxin is still there on the agar block, so for the second time exposure is not required. Okay. The third experiment performed by Auxin and Jensen, look here, already we discussed about gelatin and mica, the same thing. Okay, I'll explain it using this picture. So see the first experiment done by Darwin. I'll change the color. So first experiment was done by Darwin. So what happened when the light was passed here? What is seen? Just a minute here. So when the light was passed, what Darwin concluded, that the growth stimulus is produced in the coleoptile and is transmitted to the growth zone. Where is the growth zone? The growth zone is located below the tip region. Okay, so the Darwin says that the stimulus or the growth stimulus, which is auxin, is produced at the shoot tip and then it is transmitted to the growth zone. Clear? But when it is covered with the tip region is covered with the opaque cap, there is no bending. Okay, when the tip part is removed, then also no bending. Okay. Now in the second case, the second experiment done by Auxin and Jensen, when the gelatin block was applied, look here, when the gelatin block was put between the tip and the stump or the coleoptile, what it was seen, that the bending occurred, because gelatin is a water permeable substance. I already discussed. Now when mica sheet inserted on the dark side, no curvature, because the growth occurs in the dark side. Okay, but when mica sheet was inserted in the light region, the bending occurred, because the dark region is the region where the growth occurs. So if mica sheet is placed towards the light region, there is no effect in the bending, because the growth occurs in the dark region. Okay, so if mica is placed in the light region, then if mica is placed in the light sensitive region or in the sunny region, then there is still the bending occurred, because growth occurs in dark region. But if mica is placed in the dark region, there will be no bending. Why? Because in the dark region the cells will elongate, and in the dark region, so if this is a coleoptile, okay, and mica is placed here, okay, this is the, wait, So if I say this is a light region and this is the dark region. Okay, so if the mica is placed in the light region and the dark region is unaffected, so the bending will occur because the growth occurs in the dark region. Okay, but if mica is placed in the dark region, what we see, there will be no bending, since the dark region, since the auxin cannot diffuse through mica, or no auxin diffusion will be seen here because mica is there. Okay, no auxin diffusion will be there because mica is here, which is in water impermeable substance. So from the tip, the auxin cannot move downward. So no growth. No growth, that is no bending. Okay. The third experiment. So what is the conclusion of Auxin and Jensen's experiment? That they concluded that the stimulus can pass through gelatin but cannot pass through mica because mica is water impermeable. The third one done by Wendt, what Wendt did, that I said about the agar block. Okay, each gelatin block or agar block, whatever, each gelatin block placed on one side of the coleoptile stump, coleoptile bends in the total darkness angle to be measured. So what we see, that this gelatin block which retained the auxin, they also are able to cause the bending. Okay, I'll show you the conclusion by Wendt. But Boysen and Jensen worked with gelatin and mica, and Wendt showed that the tip region synthesizes the auxin, which is then moved to the growth zone. Paul says that the growth promoting stimulus produced in the tip was chemical in nature. Okay, that is why if you place it to any side, if you place, see, see here the experiment is already given. See in the pulse experiment here, see, if the tip part is removed, okay, the tip placed on one side, okay, so if the tip is placed on one side, the auxin concentration is more here, so the growth will be more here, so this has bent. Now see, the growth curvature develops without a unilateral light stimulus. The tip replaced on one side of the coleoptile stump. Okay, so the tip part is, from the tip part, the auxin, since it is diffusible, so even when a small part of a tip touches the coleoptile, since auxin is diffusible, auxin can diffuse to the growth zone, from the tip region to the growth zone. So the bending still occurred. So what is the conclusion of the pulse experiment? That even the place of the tip is shifted, okay, and since the auxin is diffusible in nature, they shows the bending. So this shows that the auxin stimulus is chemical in nature. Okay. Wendt shows that the growth promoting substance on diffuse can diffuse through gelatin block. That means it has first diffused from the tip to the agar or gelatin. Now from gelatin it can diffuse to the auxin, like it has happened in the pulse experiment. Now from the gelatin it is diffused to the coleoptile, okay, and showed the bending. So the graph is not important, okay. Now what is the conclusion of Darwin's experiment? That there is a tropic movement towards light. They also concluded that the light signal is perceived by the tip. And what is Boysen and Jensen's conclusion? That the signal is a mobile chemical substance which is able to move from tip to the growth zone. Since it is mobile, it is able to move from the tip, whose tip? From the shoot tip to the growth zone. Okay. Now, see, auxin can be natural or synthetically produced in industries. The synthetic auxin are 2,4-D, the common name you have to remember. The 2,4-D, the full form is 2,4-dichlorophenoxyacetic acid. Their main role is in as a herbicide. Okay, they kill the broadleaves. Okay, they act as a herbicide and they kill the broadleaves. There are some naturally synthesized auxin produced in plant named as indole 3 acetic acid, indole 3 butyric acid, 4-chloroindole 3 acetic acid, which is asked very rarely. But IPA, IBA and IAA are natural auxin which the plants synthesize in their normal conditions. Now, auxin biosynthesis. So first I'll mention the precursor was what? The precursor was a tryptophan, which is a amino acid. So we'll see several pathways of auxin biosynthesis and we'll focus on how to learn this pathway. Okay. So see the first pathway, indole 3 pyruvic acid pathway. In this pathway, what we see is the first reaction, the precursor is tryptophan, which is known to us. In the first reaction, there is a transamination reaction. This reaction is also said to be as deamination reaction. So first reaction is deamination, second is decarboxylation, and the third is dehydrogenation. This sequence you have to learn. The sequence is asked often. Okay. Now the second pathway known as a TAM pathway, there is a similarity between IPA pathway, which is indole 3 pyruvic acid pathway, and the TAM pathway. What similarity? The first two steps are reversed. In the first step of IPA pathway is deamination, and the second step is decarboxylation. But in the TAM pathway, the first step is decarboxylation and the second step is deamination. Okay, and the last step is same, which is dehydrogenation. Okay, so next another pathway is IAN pathway. In IAN pathway, what we have to study, that indole 3 acetonitrile. Full form, the explanation is given in the note, the main slides which is provided to you. In IAN pathway, what we see, that indole 3 acetyl doxine is produced first, which will produce nitrile, and then nitrilase enzyme is the one which will form IAA. So in IAN pathway, since N is there, nitrilase is the enzyme which is forming IAA. In another pathway known as IAM pathway, which is seen mainly in bacteria and Arabidopsis, the first product which is formed is indole 3 acetamide, and this, since AM is there, so it will be acetamide, which will then produce IAA. Okay, so if I summarize the whole pathway, what we see, that in the middle it was IPA pathway, where what happened, from tryptophan, what produced, the first step was deamination. In the second step, it was decarboxylation, and in the third step, it was what? Dehydrogenation. So please remember this sequence, which is very important for your exam. And the last step is IAA formation. So the name of this pathway is IPA pathway, which is the most common pathway for IAA biosynthesis in plants. So IPA pathway, it's first tryptophan, then it's deamination, then one product will be formed, then it is decarboxylation, then another product is formed, then it's dehydrogenation, then it is IAA. Okay. Now, if I go to the TAM pathway, tryptamine pathway, what we see that the first step is reversed. I have to write again. So first was deamination, second was decarboxylation, third is dehydrogenation. Okay, but in TAM pathway the first two steps is reversed. What happens in the TAM pathway is the first step is decarboxylation, second step is from tryptophan in the TAM pathway, from tryptophan it will be decarboxylation, then deamination, then it is same. Okay, decarboxylation, then through dehydrogenation it will produce IAA. Okay, another pathway which is IAN pathway, indole acetonitrile pathway. So the first product which is formed is acetaldoxine. Remember this, indole 3-acetaldoxine, which will produce one product, and through nitrilase enzyme, since N is there, through nitrilase enzyme this will form IAA. Another pathway which is common in bacteria, okay, it is seen in bacteria, and the first is acetamide. Okay, which through one more step it will form what? It will form again IAA. Clear? So this is a summary of the biosynthetic pathway, and you have to study it in this way. Okay. Now see, acetamide, then it's, so in the first two cases towards the left side the enzyme is tryptophan monooxygenase. Okay, and here it's the last, the enzyme from acetamide to acetic acid is hydrolase. Okay, so you remember first was monooxygenase in the IAM and IAN pathway to form the first product, and the last for IAM is hydrolase and IAN is nitrilase. Okay, this IAN pathway is very common in three families of plants, namely Brassicaceae, Poaceae and Musaceae. Okay, and the IAM already said they are very common in indole 3-acetamide. They are very common in bacteria named Pseudomonas and also in Agrobacterium tumefaciens. Okay.